Welcome to Curiosity, the podcast where science meets wonder. Join us as we explore the fascinating, the innovative, and the unexpected, from the secrets of the cosmos to the marvels of everyday life. Each episode takes you on a journey to uncover the knowledge and stories that fuel our endless quest for understanding. Let your curiosity guide you.
So, if you look around at the natural world, or honestly, even if you just look in the mirror over the course of a few decades,
right,
there are certain things that feel like absolute unbreakable laws of the universe. I mean, gravity pulls things down.
hot coffee gets cold.
Exactly. Hot coffee gets cold, energy dissipates, and living things get older. It just feels intuitive, doesn't it? It feels completely inevitable.
It's the standard biological trajectory.
Yeah, we're born, we grow, we reach a peak of some kind, and then slowly but surely we decline.
Which is what we see in our pets, our family members, ourselves. We call it the life cycle, right? You have development, physiological deterioration, and eventually death. And for a really long time, science just viewed this process, aging or senescence, as a universal constant.
Like a rule that just couldn't be broken.
Exactly. We thought of it as a thermodynamic necessity. Entropy applies to car engines, and it applies to biology. Things just fall apart over time.
Feels like a one-way street. You can't unscramble an egg, and you definitely can't unage a body. But, and here's where it gets really interesting and why we are jumping into this deep dive today.
Wait.
When you stop looking at humans and you start looking at the full, bizarre scope of the animal and plant kingdoms, that law of aging starts to look a lot less like and a lot more like a suggestion.
Or perhaps just a specific evolutionary strategy that some species use, but certainly not all of them.
Right, because nature is full of exceptions. And for you listening, I want to be clear about what we're doing today. In this deep dive, we are pulling from a huge array of biological case studies, evolutionary biology papers, and genetics research to understand the mission here, which is, does biology actually require us to age?
And we are going to look at some wild examples.
We really are. I don't just mean things that live a long time, like a tortoise. I mean organisms that seem to completely defy the very concept of aging as we understand it.
Microscopic polyps in freshwater ponds that might actually be immortal.
Yeah. Sharks that were swimming in the ocean for the United States became a country. Trees that have been alive since the Ice Age.
This is the whole field of biological immortality and negligible senescence.
Negligible senescence.
It's a fascinating area of study because it forces us to ask a very fundamental and honestly a somewhat uncomfortable question.
Which is?
Is aging truly inevitable? Is it a strict requirement of being alive? Or is it a biological problem that evolution has actually already solved in other contexts and we just haven't figured out how to access that solution yet?
So today, we are going to unpack this biological paradox. We'll look at the definitions, because the words really matter here. We'll get into the evolutionary theories of why we age, like why nature lets us fall apart in the first place.
The selective pressures.
Exactly. And then we're going to do a massive review of the specific creatures, these rebels of the biological world that have managed to decouple time from physiological decay.
And just to set the scope here for you, as we always do, we aren't talking about magic.
No vampires.
Right. No science fiction or the literal fountain of youth. We are looking at specific evolutionary and cellular mechanisms. We want to understand how these organisms work on a molecular level because understanding them is the key to understanding the limits, or maybe the lack of limits, of aging itself.
Okay, let's start with the absolute basics. We use the word aging all the time. Like, I'm aging, this cheese is aging in my fridge.
Hopefully not the same way.
Yeah, hopefully not. But in a scientific context, we need to be much more precise. We need to distinguish between just existing for a long time and the actual process of biological decline.
Precisely. This is the core distinction between chronological aging and senescence.
Okay.
Chronological aging is simply the passage of time. It's a measurement on a clock or a calendar. If a rock sits on a hill for 1,000 years, it is chronologically aged. It is older today than it was yesterday.
But it hasn't senesced.
No, it hasn't senesced in the biological sense. Senescence is a very specific biological process. It is the progressive deterioration of physiological function. It's the breakdown of the actual machinery of life.
So when we talk about senescence, what are the actual metrics? How do you measure it?
We are looking for a few specific things. A decrease in physiological efficiency, a decline in the body's repair mechanisms, and, most critically, an increase in what we call the hazard rate.
The hazard rate. That sounds incredibly ominous. Let's unpack that for a second.
It is the main statistic that biogerontologists look at first. The hazard rate is essentially the probability of death at any given age.
Okay.
In most organisms, including humans, the hazard rate increases over time. This is known mathematically as the Gompertz law.
So practically speaking, my chance of dying today as a healthy adult is pretty low. But my chance of dying on a specific Tuesday when I'm 95 is much, much higher.
Correct. The longer you live, the more frail your systems become, and the higher the daily probability that your system will experience a catastrophic failure. That exponential increase in the risk of death, that curve going up, that is the signature of senescence.
But you mentioned earlier, and this is in our sources, there is this concept called negligible senescence, which linguistically sounds like an oxymoron. Negligible aging.
It does sound impossible, but it is a recognized, documented biological phenomenon. The term was actually introduced by the biogerontologist Caleb Finch back in 1990.
Caleb Finch, yeah.
He needed a term to describe organisms that simply do not follow that Gompertz law. They don't have that upward curve.
So what is the strict operational definition here?
An organism is considered negligibly senescent if it demonstrates no measurable increase in its mortality rate and no decline in its reproductive capability as it gets older.
Wait, let me make sure I truly understand that. If an organism exhibits negligible senescence, a 100-year-old individual is no more likely to die tomorrow than a 5-year-old individual.
That is exactly it. The baseline risk of death remains constant. It doesn't curve upward. And typically, their ability to reproduce doesn't drop off either. A 100-year-old individual is just as fertile, sometimes even more fertile, because they are larger than a young one.
That is absolutely mind-bending. It goes against literally everything we experience as humans. We are so used to the idea of a prime of life, followed by a long decline. But these creatures just stay in their prime.
Indefinitely, yes. However, we need to make a very crucial distinction right now so we don't confuse the issue. When we use the term biological immortality or negligible senescence, we are not talking about absolute immortality.
Right. They aren't invulnerable. Superman is immortal. These things are just, well, something else.
Correct. An organism with negligible senescence is not invulnerable to extrinsic factors.
Extrinsic meaning outside stuff.
Yes. A predator can still eat it, a disease can still kill it, a sudden change in water temperature or a drought can wipe it out. If you step on it, it dies.
So if a piano falls on a biologically immortal creature, it's still flat.
Exactly. Biological immortality simply means it lacks an intrinsic aging mechanism. It doesn't have an internal expiration date programmed into its cells. It won't die of old age.
Because old age isn't a factor.
Right. If you could protect it from all those outside threats, the predators, the starvation, the accidents, theoretically, it could keep going indefinitely.
Okay, that is a powerful concept. So if this is possible, if biology allows for organisms to just not fall apart over time, the big question, the one that I think you listening are probably screaming at your device right now is, why don't we do that?
That's the obvious question.
Right. Why has natural selection allowed the costs of deterioration and death to persist in so many species, especially us? Why haven't we evolved this forever young trait if the biology is clearly capable of it?
This brings us to one of the most elegant and somewhat counterintuitive areas of evolutionary biology. It forces us to rethink what evolution actually selects for.
Because we think of evolution as wanting to make the best, strongest thing.
Right. You might think evolution would want to create the perfect, long-lasting organism, an immortal super being. But evolution doesn't actually care about longevity. It cares about genetic transmission.
It cares about passing on the baton. It doesn't care how long the runner lives after the handoff.
That is a perfect way to put it. Once you have passed on your genes and ensured your offspring can survive, from the cold perspective of natural selection, you are largely expendable.
Brutal.
It is. And there are a few key theories that explain how this plays out. The first is the mutation accumulation theory, which was proposed by Peter Medawar in the 1950s.
Okay, how does mutation accumulation explain why we get wrinkles and our joints give out?
Medawar realized that the strength of natural selection weakens drastically as an organism gets older. Think about an animal in the wild. A mouse, for example. The environment is incredibly dangerous. Predators, cold weather, starvation, accidents. Most mice die very, very young.
Right. A mouse in the wild probably gets eaten by an owl way before it ever develops heart disease or arthritis.
Exactly. The odds of a wild mouse surviving to age 3 are astronomically low. So imagine a genetic mutation arises in the mouse population. And let's say this mutation causes a terrible, fatal neurodegenerative disease, but the disease only activates when the mouse reaches age 4.
But no mouse ever reaches age 4 because of the owls and the cold.
Precisely. Because almost no individual lives to that advanced age, natural selection cannot see that mutation to weed it out. The mutation doesn't affect the mouse's ability to run away or reproduce in its prime at 6 months or a year old. So the mutation just gets passed on.
It drifts through the gene pool.
Yes. It's like a terrible flaw in a car's engine that only happens after 1,000,000 miles. The manufacturer doesn't bother fixing it because no car ever actually goes that far in the real world.
So these mutations just hide.
They do. These late-acting deleterious mutations accumulate in the genome simply because nature doesn't scrub them away. They pile up in what we call the shadow of selection. And when we, as humans, started living longer due to modern safety and medicine and agriculture, we basically slammed right into this invisible wall of accumulated genetic junk.
That makes total sense. It's benign neglect. Nature just stops watching after a certain point.
Yeah.
But there's another theory we found in the research that suggests it's not just neglect, but actually a deliberate trade-off.
Yes, the antagonistic pleiotropy hypothesis.
Antagonistic pleiotropy. Try saying that 3 times fast.
It's a mouthful. This was championed by the evolutionary biologist George Williams, and it's a bit more active than Medower's neglect theory. Pleiotropy refers to a single gene having multiple effects on an organism. Williams suggested that some genes might be antagonistic, meaning they have opposing effects at different times in the life cycle.
So a gene could be really good for you at one point and actively bad for you at another.
Exactly. Imagine a gene that heavily boosts your cellular growth and reproductive hormones when you are young. It makes you super fertile, strong, fast to heal. It helps you outcompete rivals, find a mate, and have lots of offspring.
That sounds like a huge evolutionary advantage.
It is. You win the evolutionary game in the short term. But let's say that exact same gene, that same high level of cellular growth signaling, causes tissue damage or fuels cancer growth when you are 70.
Oh, I see. So you trade longevity for early fertility.
And because nature desperately prioritizes early fitness, ensuring you survive long enough to reproduce at all, those genes are aggressively selected for. The late life damage is just the price paid for early life success. We are essentially programmed to burn bright and fade out quickly, rather than burn slow and last forever.
This connects perfectly to the idea of extrinsic mortality you mentioned earlier. The environment plays a huge role in determining these biological trade-offs, doesn't it?
It provides the entire context for the trade-off. The theory predicts that organisms with high natural defenses against those extrinsic hazards, the things that keep them safe from predators, should naturally evolve slower aging rates over time.
So if you have armor or weapons or a really good hiding spot, your species can afford to play the long game.
Right. Think about it logically. If you are a standard mouse, you are going to die fast no matter what, so you have to evolve to reproduce as fast as possible, and consequently, you age fast. But if you have a hard shell, like a turtle, or you can fly away from danger, like a bat or a large bird, your daily chances of surviving increase significantly.
The owl can't eat you if you are safely tucked inside a hard shell.
Exactly. And suddenly, over evolutionary time, it becomes highly profitable to maintain your body for longer. The warranty period extends. The selective pressure to repair your DNA and maintain your tissues increases because there is actually a good statistical chance you will be around to use those tissues in 50 years.
That logic holds up so well when we look at the animal kingdom. Birds, bats, turtles, they routinely live much longer than similar-sized ground animals that are more vulnerable.
It's a very consistent, observable pattern. Low extrinsic mortality strongly correlates with the evolution of longevity. And this brings us right to our case studies, because we have these theoretical frameworks, but the real fascinating part is seeing how nature has actually implemented them.
Let's start with the absolute poster child of negligible senescence. The organism that seems to have just thrown the rulebook entirely out the window, the Hydra.
The Hydra is a deeply fascinating creature to study. It's a small, freshwater invertebrate. It's basically a tiny translucent tube with a sticky foot at one end to hold onto a rock and a ring of tentacles at the other end to catch food. It belongs to the phylum Anaria.
So it is related to jellyfish and sea anemones.
Yes.
And when we say this thing doesn't age, we have actual hard data on this, right? It's not just some biologist's anecdote from a pond.
We have very rigorous data. There were landmark studies conducted by a researcher named Daniel Martinez, first published in 1998, and then a massive follow-up in 2012. He kept large populations of Hydra in his lab and tracked every single individual for years. 4 years in the first study and 8 years in the second.
Now, for a tiny invertebrate that basically looks like a piece of thread, 8 years is an absolute eternity.
It is a vast lifespan for an animal that size, and in all that time, he monitored them daily. He looked for any signs of decline, and he found literally nothing. Constant mortality rates. The risk of death did not go up by a fraction of a percent. A Hydra that was 8 years old was just as spry and capable of catching food as a Hydra that was 2 weeks old.
And their reproduction?
Their reproductive output. And they reproduce by budding, which is basically growing a mini version of themselves off their side that pinches off, that didn't decline either.
So how do they do it? What is actually happening at the cellular level? Is it just constant repair?
It comes down to stem cells. To understand the Hydra, you have to understand how fundamentally different it is from us. Most animals, humans included, have a lot of what we call post-mitotic cells.
Post-mitotic, meaning after division.
Right. Your brain cells, your heart muscle cells. They divide rapidly while you're developing in the womb, and then they mostly stop. They differentiate, they take on their specific job, and eventually they wear out, sustain damage, or die. And because they don't divide anymore, they are incredibly hard to replace. When they are gone, they are gone.
But the Hydra doesn't play by those rules.
Not at all. The Hydra lacks permanent, non-dividing tissue entirely. Its entire body is composed of 3 distinct populations of continuously dividing stem cells. It is constantly, endlessly replacing its entire cellular content.
It really is the biological version of the ship of Theseus paradox. If you replace every single wooden plank on a ship over time, is it still the same ship? If the Hydra replaces every single cell in its body every few weeks, is it the same Hydra?
Functionally and genetically, yes. It maintains its physical pattern and its life. It avoids senescence by a strategy of constant total renewal. It simply never lets any of its cells get old enough to fail. And we have actually identified some of the genetic machinery involved in keeping those stem cells so incredibly active.
This is where we get to the specific genes.
Yes. The FOXO transcription factor.
FOXO. I feel like I've heard of that in human anti-aging research, too.
You definitely have. It's a highly conserved gene across the animal kingdom. We have it, microscopic worms have it, fruit flies have it. It's a master regulator gene. It acts as a switch that controls dozens of other genes involved in DNA repair, stress resistance, and regulating the cell cycle. In the Hydra, FOXO is expressed at extraordinarily high levels in all 3 of those stem cell lines all the time.
So it's basically keeping the cellular repair crews working overtime, 204/7.
Exactly. And researchers proved this by doing a genetic knockdown. They experimentally reduced the expression of the FOXO gene in a living Hydra, and the result was immediate. The stem cells started to lose their potency, they stopped dividing effectively, and the animal actually started to show classic signs of aging. It deteriorated. So FOXO is a critical, irreplaceable piece of their immortality puzzle.
Okay, so the Hydra stays young by never letting its cells get old. It's a strategy of relentless turnover. But there is another creature in our sources that takes this a step further. Instead of just maintaining the status quo, it can actually hit the rewind button.
Yes!
The immortal jellyfish.
Turritopsis dorni. This is a biological anomaly that borders on the unbelievable. This specific species of jellyfish has the capability to revert from a fully sexually mature adult stage, which is called the Medusa stage, all the way back to its juvenile polyp stage.
That is incredible. Just to put that in human terms for a second, that would be like a 40-year-old human deciding, you know what, taxes and back pain are too stressful, and just physically turning back into a toddler.
That is a highly accurate analogy. And interestingly, it usually happens in response to specific environmental triggers. It's not necessarily just a standard part of a continuous life cycle loop. It's a dedicated survival mechanism. If the adult jellyfish is physically injured, or if it is starving, or if the water temperature or salinity becomes too stressful, it triggers this developmental reversal.
So when things get tough, it retreats to childhood. I think we can all relate to that impulse on some level. But biologically, how do you turn a complex swimming adult body back into a simple, stationary juvenile one?
The cellular mechanism is called transdifferentiation.
Let's unpack that term, transdifferentiation.
Usually, cellular development is a strict one-way street. A blank stem cell differentiates into a muscle cell or a nerve cell. Once it commits to that path, it's committed. It can't go back. Transdifferentiation breaks this fundamental rule. It is a process where a highly specialized, mature somatic cell actually reverts to a totipotent stem-like state.
It essentially forgets what it was.
It completely resets. It loses its specialized structures and becomes a basic building block again. And then it redifferentiates into a completely different cell type that is needed for the juvenile polyp stage.
It completely resets the developmental clock.
Completely. The adult medusa literally dissolves its own tentacles in its bell. It sinks and settles onto a hard surface on the seafloor and transforms back into a blob of tissue that becomes a polyp colony. And from that new colony, fresh, genetically identical jellyfish are eventually released back into the water.
So it's not just saving the individual, it's actually cloning itself in the process. But again, ecological context matters here. In the wild ocean, they don't actually live forever, do they?
No, they don't. They're tiny bits of plankton. They get eaten by almost everything. Small fish, sea turtles, other larger jellyfish, and they are highly susceptible to ocean diseases. This reversal process is an amazing stress response, but it doesn't protect them from being swallowed whole by a passing fish.
So we don't have an ocean currently overflowing with billion-year-old jellyfish.
No, but theoretically, the intrinsic potential for infinite life cycles is there. If you kept one in a perfectly safe, controlled laboratory tank and stressed it just right, it could loop back and forth indefinitely.
Okay, so those are the squishy, weird invertebrates. Let's talk about something a bit more substantial, something with a shell. We have to talk about the ocean quahog.
Arctica elendica. This is a species of edible clam that lives in the very cold, deep waters of the North Atlantic Ocean. And it currently holds the record for the oldest non-colonial animal ever discovered.
You're talking about Ming.
Yes, Ming the clam. This is a simultaneously tragic and amazing scientific story. Back in 2006, researchers were dredging up these clams off the coast of Iceland. They were actually trying to study historical climate change. They wanted to analyze the shells.
Because clam shells have growth rings, just like trees.
Exactly. They put down a new microscopic layer of shell every single year. By analyzing the oxygen and carbon isotopes trapped in those specific rings, you can essentially read a diary of ocean temperatures going back hundreds of years. So the researchers collected these clams, froze them on the ship to preserve them, and took them back to the lab. And it was only when they opened one particular, very large clam to count the rings under a microscope that they realized what they had done.
They accidentally killed the oldest known animal on Earth.
By painstakingly counting the rings, they confirmed its age at 507 years old.
507 years. That means this specific clam was born in the year 1499. That is just mind-boggling to think about. It was sitting silently at the bottom of the freezing ocean while Leonardo da Vinci was painting the Mona Lisa. It was there during the entire Reformation, the American Revolution, the Industrial Revolution, 2 world wars, all the way to the age of the smartphone.
It is profound. And interestingly, the ocean quahog doesn't fit the strict definition of negligible senescence perfectly. It does age, but it ages incredibly slowly. Its mortality rate stays extremely low and remarkably flat for centuries before finally ticking up.
So what is its secret? Is it really just the cold water slowing everything down?
The cold environment certainly helps. It severely slows down their overall metabolic rate. But it's definitely not the whole story. The quahog seems to be an absolute master of biochemical defense. Studies on their tissues show they have vastly superior antioxidant defenses compared to short-lived clams. But perhaps most importantly, it has incredibly high-efficiency protein quality control.
Protein quality control. We hear about proteins all the time in nutrition, but why is the quality control of them so critically important for the aging process?
Because the accumulation of misfolded proteins is a primary hallmark of aging and cellular disease. Proteins are very complex, three-dimensional shapes. If they fold into the wrong shape due to stress or errors, they don't do their job. Worse, they get chemically sticky, they clump together. In human diseases like Alzheimer's and Parkinson's, we see exactly this. Protein aggregates, which are essentially clumps of junk protein, building up inside neurons and eventually killing the cells.
It's like trash piling up inside a house. Eventually, you literally can't move or function.
Exactly. And most organisms, as they get chronologically older, their cellular machinery gets worse and worse at taking out that trash. The molecular sweepers break down. The quahog, however, does not. It is incredibly good at maintaining what we call proteostasis, which is protein stability. It actively keeps its molecular house clean for centuries. It relentlessly prevents those toxic aggregates from ever forming in the first place.
That is a perfect segue to the vertebrates, because usually when we talk about extreme longevity in animals, We're looking at cold-blooded things like clams or jellyfish or sharks. They have slow, plodding metabolisms.
Yeah.
But there is a mammal, a warm-blooded, high-energy, running-around mammal that breaks all the rules we just established.
Yes.
The naked mole rat.
The naked mole rat is truly the great mammalian anomaly. If you look at standard metabolic scaling laws, there is a very reliable general rule in mammals. Big animals live a long time, small animals die young, an elephant lives 70 years, a tiny field mouse lives maybe 3.
High heart rate, live fast, die young. And a naked mole rat is roughly the exact size of a mouse.
Right. By all the known laws of mammalian biology, it should live about 3 years. But in captivity, they can live more than 30 years. That is a massive tenfold difference.
Yeah.
If a human had that kind of biological scaling advantage, we would routinely live to be 800 years old.
And it violates the Gumpert's law of mortality we talked about at the beginning, right?
This is the absolute key finding that put them on the map for gerontologists. A major 2018 study published in the journal A Life analyzed thousands of historical data points from mole rat colonies kept in labs around the world. They found that after they reach sexual maturity at about 6 months old, their daily probability of dying remains entirely constant. They simply do not show that exponential upward curve in death risk that characterizes humans, mice, and virtually every other mammal.
So they are, for all intents and purposes, biologically negligible senescent mammals.
It appears so based on the math. A 20-year-old mole rat has the cardiovascular health, the bone density, and the reproductive capacity of a 2-year-old mole rat.
Their physiological adaptations must be just incredible. We talked about protein quality control with the quahog clam. Do mole rats do that too?
They do. They are also champions of proteostasis. They degrade and clear out misfolded proteins much faster and more accurately than mice do, but they also have legendary, almost unbelievable cancer resistance.
I've read a bit about that. Spontaneous cancer is almost unheard of in naked mole rats.
In decades of intense observational study involving thousands of animals across multiple research centers, only a tiny handful of cancer cases have ever been documented. And one of the primary mechanisms behind this involves a very specific molecule called high molecular mass hyaluronic acid.
Hyaluronic acid, wait, that's in skin creams. It's a very common beauty product.
It is. In humans, it's a component of connective tissue that helps keep skin hydrated and plump. But the version produced by naked mole rats is chemically different. It is much, much larger. It's a high molecular mass polymer, and it is secreted to be 5 times more abundant in their tissues than in ours. It's actually what gives their skin that highly distinctive, loose, baggy, stretchy quality they are famous for.
Which I assume helps them squeeze through tight underground tunnels without tearing their skin.
Right, that's the mechanical benefit. But scientists discovered it also fundamentally regulates their cellular behavior. This incredibly dense, sugary goo packed around their cells acts like a physical cage. It forcefully triggers a phenomenon called early contact inhibition.
Explain early contact inhibition. What does that mean for cancer?
Normal, healthy cells stop dividing when they physically touch other cells. They know there isn't room. Cancer cells, by definition, lose this inhibition. They ignore the crowding, they pile up on top of each other, and they form dense tumors. The massive hyaluronic acid molecules in mole rats make their cells incredibly sensitive to crowding, way more sensitive than our cells. As soon as mole rat cells start to get even slightly too dense, the cell cycle forcibly arrests. It physically and chemically shuts down the uncontrolled growth required for a tumor to even begin forming.
That is absolutely fascinating. It's a literal molecular safety net built into their skin and organs. And we should probably mention their lifestyle because it ties back to the evolution. They live entirely underground in highly complex eusocial colonies, much more like ants or bees than other mammals.
Which brings us perfectly back to the evolutionary theory of aging we started with. Why did they, of all mammals, evolve this extreme longevity? Because they are incredibly protected from extrinsic mortality. They live deep in hard, baked clay tunnels in East Africa. They have a highly organized queen and worker social structure. They are fiercely defensive of their burrows.
The tunnel network essentially acts like the turtle's shell.
Exactly. It drastically, permanently reduces extrinsic mortality. They aren't getting picked off by hawks or wildcats every day. This immense safety provided the evolutionary space, the deep time, for them to slowly develop these complex, energy-intensive longevity mechanisms. They didn't have to rush to reproduce in a few months.
Okay, Moving on to some other notable vertebrates, we have to mention the Greenland shark. If the mole rat is the undisputed king of rodents, this thing's the king of the ocean.
The longest-lived vertebrate on Earth. For a very long time, marine biologists didn't actually know how old they were. They are cartilaginous fish. They don't have hard calcified bones like other fish, so you can't just cut a bone and count the growth rings.
So how did we finally figure out their age?
Radiocarbon dating of the lens of their eye.
The lens of the eye. That seems incredibly specific.
Yes. The very center of the eye lens, the nucleus, is formed when the shark is just a tiny embryo. It consists of highly specialized, transparent proteins that are metabolically completely inert. They do not turn over. They do not change. Ever. It's a perfect biological time capsule from the exact moment of the shark's birth.
And because humans detonated lots of nuclear weapons in the atmosphere in the 1950s, we accidentally put a massive spike of a specific carbon isotope, carbon-14, into the atmosphere and the ocean.
Exactly. That nuclear bomb pulse acts as an undeniable timestamp in the biological record. By analyzing the carbon isotopes trapped in the embryonic eye lenses of 28 female Greenland sharks caught as bycatch, researchers were able to calculate ages up to roughly 400 years.
400 years old.
And here is the real kicker about their biology. They grow so incredibly slowly that they don't even reach sexual maturity until they are about 150 years old.
Imagine being an awkward teenager for a century and a half. That sounds terrible.
It suggests an incredibly, almost unfathomably slow metabolic pace. Everything about their life history is stretched out to the absolute extreme. They swim slowly in freezing water. Their hearts beat very slowly. They grow at a glacial pace, maybe about 1 single centimeter a year.
It's the ultimate live slow, die old strategy.
Deep sea cold storage.
And just quickly, before we move out of the animal kingdom and into plants, let's briefly touch on rockfish and tortoises, because the rockfish data is really revealing regarding genetics.
Rockfish, which are in the genus Sebastes, are fantastic for genetic research because you have highly closely related species living in the same oceans with vastly different natural lifespans. The calico rockfish, for example, lives about 10 years, but its close cousin, the rough-eye rockfish, lives well over 200 years.
That's a huge, huge difference for genetic cousins.
It is, and it allows for very precise comparative genomics. We can line up their entire DNA sequences next to each other and ask the computer what is the exact difference. It turns out the long-lived rockfish species have massively expanded gene families controlling DNA repair and inflammation regulation. They literally just have more tools in their genetic toolkit to fix daily cellular damage.
In tortoises, of course, we can't skip the classic example.
The Aldabra and Galapagos giant tortoises, we know they can easily exceed 150, maybe 200 years. Their mortality patterns are highly consistent with negligible senescence, although statistically it is very hard to prove definitively, simply because we don't have enough rigorous historical data on 200-year-old tortoises to build a mathematically robust Gompertz curve. Humans just don't live long enough to track them properly across multiple generations.
We die before the experiment is over. Okay, let's completely shift gears to the plant kingdom, because we are talking about really old living things. Animals are honestly just amateurs. Trees are the undisputed heavyweights. But here the actual definition of what an individual is gets a little tricky, doesn't it?
It gets very philosophical. We have to clearly distinguish between a single physical stem, what botanists call a ramette, and the actual underlying genetic organism, which is called the genus. This brings us to Pando.
Pando, also known as the trembling giant.
It's an absolutely massive colony of quaking aspen trees located in Utah. To the naked eye, if you just walked through it on a hike, it looks like a completely normal forest of about 40,000 individual separate trees. But extensive genetic testing shows they are all completely identical clones. They are all physically connected underground by a single, sprawling, massive root system.
So it's technically one single giant organism.
One genetic individual. And while the individual stems, the actual wooden trees you see above ground, might only live for maybe 100 or 130 years before getting a disease and falling over, the massive root system just keeps endlessly sending up fresh new shoots. The entire organism itself, the genus, is estimated by some to be around 80,000 years old.
80,000 years?
Yeah.
That's deep back in the Ice Age. It was slowly growing in Utah when early humans were just starting to migrate out of Africa.
It is essentially immortal as long as the local geological and climate conditions allow it to keep growing. It survives through vegetative reproduction. It has completely decoupled the long-term survival of its genetic unit from the temporary survival of any single wooden trunk.
But there are also non-clonal trees that live a staggeringly long time. The bristlecone pines. These aren't connected clones.
No. These are single, distinct, isolated individuals. The most famous documented specimen is named Methuselah, located high up in the White Mountains of California. Core samples indicate it is about 4,850 years old. It literally germinated from a seed around 2800 BCE.
That is older than the pyramids of Giza. It was a sapling when they were building Stonehenge.
It is, and what's amazing is that even at that extreme age, these trees actively maintain photosynthesis. They still produce perfectly viable seeds. Their pollen is tested to be just as genetically sound as a tree that is only 50 years old. They actively resist disease.
How? I mean, they just stand there on these freezing dry, incredibly windy mountain ridges, getting blasted by intense UV radiation for 5 millennia. How do they not just turn to dust?
It's a combination of extreme, harsh environmental adaptation. First, they have a very, very slow metabolic rate, dictated by the cold and dry conditions. Second, their physical wood is incredibly dense and heavily resin-rich. It acts as a powerful, natural chemical preservative against boring insects and fungal rot. It's almost as hard as stone. But they also use a brilliant physiological strategy called sectorial integration.
Sectorial integration. What exactly is that?
Think about a mammal. Most animals are fully integrated systemic organisms. If your heart stops, your brain quickly dies. If your kidneys fail, your blood becomes highly toxic and every single cell dies. We are built like a fragile house of cards. A tree like the ancient bristlecone pine is built more like a highly compartmentalized ship.
Like a military submarine with those watertight doors that seal off sections.
Exactly like that. In a bristlecone, a specific underground root connects only to a specific narrow strip of bark on the trunk, which in turn only feeds a specific set of branches in the canopy. If a root gets exposed and dies from drought, only that one specific connected strip of the tree dies. The rest of the tree is physically and functionally completely unaffected. It allows them to survive and persist, even when up to 90% of the physical tree mass is technically dead wood.
So you look at Methuselah, and it looks mostly like a twisted dead stump with just one tiny green branch. But that one green branch is perfectly biologically healthy.
Correct. It continually sacrifices the damaged parts to save the functional whole.
I want to touch briefly on the microbial world before we try to synthesize all this crazy information, because this is where the philosophy of aging gets really weird. Bacteria divide. They don't have sex, they just split in two. So does a bacterium ever actually die of old age?
This is a deep, ongoing question in microbiology. For a very long time, scientists genuinely thought simple binary fission was a form of true biological immortality. One cell grows, splits down the middle, and becomes two fresh young cells ad infinitum. But highly advanced microscopic research on E. coli has recently shown something called asymmetric aging.
Asymmetric aging.
Imagine a standard rod-shaped bacterium. When it grows and divides, it builds a new cell wall right in the middle and splits apart. The newly formed ends at the split are what we call the new poles. The old ends at the far outer sides are the old poles.
So every time it divides, every new bacterium has one brand new end and one older end.
Right. But when those two daughter cells go on to divide again, one of them inherits the original older pole from the grandmother cell. It's essentially a biological hand-me-down. Researchers actually painstakingly tracked these specific lineages under microscopes for hundreds of generations and found that the specific cell line constantly inheriting that old pole physically grows slower, accumulates far more toxic protein aggregates, and is statistically much more likely to eventually die.
So even in single-cell bacteria, somebody gets left holding the biological bag.
It certainly seems so. Nature actively segregates the accumulated cellular damage. It intentionally dumps the metabolic trash into one specific lineage so the other lineage can be born completely fresh and undamaged. It strongly suggests that even the most simple, single-celled life forms have to invent strategies to deal with entropy and structural damage accumulation over time.
Okay, so we've looked at jellies that reverse time, clams that live half a millennium, cancer-proof mole rats, trees that outlast civilizations, and bacteria passing down trash. When we step back, when you look at the big picture of all these sources, what actually connects these dots? Are there universal biological rules for living a long time?
There absolutely is a clear phenomenon of convergent evolution happening here. These species are not closely related at all. A clam and a pine tree are wildly different branches of life. Yet they have all independently settled on very similar biological solutions to solve the fundamental problem of time. Based on the literature, I would identify 4 core, non-negotiable requirements for negligible senescence.
Okay, let's list them out. What's the first one?
First is genome integrity. You absolutely need superior DNA repair mechanisms. If you can't constantly fix the genetic blueprint when it gets damaged by radiation or metabolism, you cannot last. The rockfish and the Hydra show us this perfectly.
That makes total sense. If the blueprint rots, the building falls down.
Exactly. Second is proteostasis. We keep coming back to this word. You must have a robust, relentless system to clean up misfolded proteins and cellular waste products. The quahog clam and the naked mole rat are the undisputed masters of this. You simply have to take out the molecular trash.
Relentlessly.
Third is stem cell potency. You need an active, protected way to continuously renew tissues to replace the cells that are inevitably lost or damaged. The Hydra is the ultimate extreme example of this, but even humans rely heavily on this to maintain our gut lining and blood over our shorter lifespans.
And the final pillar.
Stress resistance. You need incredibly effective antioxidant defenses to handle the harsh chemistry of life without literally rusting from the inside out over the decades.
And this brings us to the ultimate, so what for the listener today. Why does this deeply weird zoological knowledge matter for human biomedical research? I mean, we aren't going to turn into giant clams or subterranean mole rats.
No, we certainly aren't going to start injecting ourselves with jellyfish juice to try and turn back into toddlers. But this research proves a massive fundamental point about biology. Aging rates are incredibly malleable. They are not fixed, immutable constants of physics like the speed of light or gravity. They are flexible evolutionary variables.
They are dials that can theoretically be tweaked.
Exactly. And we're increasingly finding that the underlying genetic pathways controlling those dials are highly conserved across species. That FOXO signaling pathway we saw acting as a master switch in the Immortal Hydra, humans have that exact same gene. The proteostasis mechanisms keeping the clams clean, we have versions of those enzymes in our cells. The major regulatory metabolic pathways we study in longevity labs like mTOR, sirtuins, the insulin-IGF-1 axis, these are all present and active in humans.
So the goal of modern geroscience isn't to weirdly transplant a naked mole rat gene into a human, but to deeply understand how their hyper-efficient version of the pathway works, and maybe figure out a pharmaceutical or genetic way to tune our own existing pathways to work a bit more like theirs.
Precisely. It provides a literal biological roadmap. It definitively shows us that physiological deterioration is just a complex biological problem, and nature has already engineered specific, observable solutions to it. It completely shifts the medical paradigm from, how do we aggressively treat the late-stage symptoms of old age, like trying to cure advanced cancer or treating heart disease individually after they happen.
Playing whack-a-mole with diseases.
Exactly. It shifts it to, how do we address the root cellular cause of senescence itself?
Because if you can just maintain the machine properly, the parts don't ever break in the first place.
And if you can maintain the stability of the genome and the proteome at a cellular level, the devastating diseases of aging we fear so much might simply never arise. That is the incredible promise of the field of geroscience.
So we have traveled all the way from the seemingly inevitable depressing pull of gravity to the complete biological defiance of the Hydra. We've seen that while humans might feel trapped on a one-way downward escalator of aging, nature has actually built elevators, endless loops, and biological pause buttons all over the place.
The mere existence of these wildly different organisms definitively demonstrates that biological deterioration is not a strict thermodynamic necessity of life. It is an evolutionary choice, or at least a specific evolutionary outcome that can, in theory, be modified.
Which leaves you with a really provocative thought to end on today. If a simple, humble jellyfish floating in the ocean can figure out how to literally reset its own developmental clock, and a blind, hairless mole rat can figure out how to stop the calendar from destroying its cells underground, what are the actual fundamental limits of human potential? Are we really hitting a hard, unbreakable biological wall at around 100 years, or have we just not figured out the right genetic code to input yet?
That is the exact question that will undoubtedly drive the entire next century of biological research. And honestly, looking at the sheer pace of the progress we are making right now, I wouldn't bet against us eventually figuring it out.
And on that note, we'll leave you to ponder your own biology. Thanks for joining us on this deep dive.
It's been a real pleasure.